Hello dear friends. So on today we are going to discussing about the structural similarities which are found into sequences or more than two sequences, that are domains, motifs, profiles, patterns and fingerprints. So these are the structural similarities. These are stored in the protein secondary sequence databases, as you know that. So all these are super secondary structures. So on today we are going to discuss about all these points. So let's revise little bit what we have already learned. So there are four structural levels of protein. The primary structure, you know that it is a linear chain of amino acid, one can call it as a sequence. So that sequences of amino acids are stored in primary protein sequence databases. When the sequence is get fold into proper structure, that is alpha helix or beta sheet, that is stored in the secondary databases. But that folding pattern or the sequence, a specific sequence present in between amino acid, that is called as motif. If you look out this A, S, then I, L, 2, X, D, E, R, F, Y, W, 2, H. So when multiple sequence alignment is carried out, specific pattern which is conserved in each and every amino sequence, so that sequence is called as motif. We will going to learn what is the motif, what is its significance, what are the similarities between domain and motif in upcoming slides. Okay, so these motifs are the super secondary structure which are present in the protein sequence. Then more than one motif forms a domain, and more than one domain form a tertiary structure, and tertiary structure is nothing but three-dimensional structure which is stored in the structural databases. So these are the levels of sequence and structural organization in case of protein. So these are the levels of sequence. So the random sequence, then specific sequence one can call it as motif, specific many sequence one can call it as a domain. Okay, and entirely primary sequence, secondary sequence and tertiary sequence. So these are the different types of databases which stores the data. So let's discuss the motif concept. So these are the multiple sequences. Suppose this is the first, second sequence, third, fourth and 5 sequences are there, and these blackish regions are the common regions in each and every sequence. So this single region is called as single motif, that is it has, suppose this is C, Y, D, E, G, G, I, S. This is the sequence where first letter C is common to all, second Y is common to all, but third letter there is a different D, E, E, H or R. But next E and D, only E and D is there, then G and D. So this is a specific type of sequence which is found in each and every sequence. If only single motif is considered, so it is a single motif is considered, such motif are stored in this database, that is ProSite. As you learn that there are different types of protein secondary sequence databases: ProSite, Profile, PPAM, Prints, Blocks and Identity. So among this is the ProSite database which stores the single motifs, and after that these are stored in the regular if this expression occurred in regularly, that is stored in the identity database. If multiple domains are taken, suppose this is the first domain, this is the second domain, second, then third and fourth. If multiple domains are taken, then they are stored in the prints database, and after that they are weighted, the specific matrices is applied on that database and that are stored in the blocks database. If the entire sequences are considered, that is full domain alignment methods are used, then one can store that into profile library, or one can also store it into the PPAM. So these domains or motifs are related with the local sequence alignment. You have studied the sequence alignment. There are two methods: local sequence alignment, global sequence alignment, pairwise sequence alignment and multiple sequence alignment. So these motifs profiles are related with the multiple sequence alignment, but in multiple there are local sequence alignment, and if the global sequence alignment is carried out, then they are stored in the hidden Markov models. So let's discuss what is a motif. I have already discussed that it is a consecutive string of amino acids in a protein sequence, okay, whose general character is repeated or conserved in all sequences in a multiple alignment at a particular position. So motifs are of interest because they may correspond to the structural and functional elements within the sequence they characterize. So let's see. So this is a motif and this is this sequence. So this is the first C, second is the X, second is the Y, which is common to all, third is the X2. So X means any two amino acid, X is the any amino acid. Again DG, so DG is common to this sequence, either D or either G. Again this is a G common, and this is a X means any amino acid at this position, again S or T. So this is regular expression, one can also call it as a pattern, and specific pattern within all sequence is called as motif. So consecutive string of amino acids in a protein whose general character is repeated or conserved in all sequences, which is called as motif. So this is all about motif. Regarding pattern, you will learn in the upcoming slide. So fingerprint is also there, we will learn in the next slide. So again, it is a particular arrangement of secondary structure that can be found in proteins called as motif. So short segment of 3D structure, spatially close but not necessarily adjacent to the sequence. So motif is a connection of helices and sheets in a different combination. The motif contain either helix turn helix or helix loop helix or hairpin model. So you have studied the secondary structure, that is either alpha helix if it is left-handed or right-handed, otherwise the beta sheet. It is parallel or anti-parallel hydrogen bonding, that is parallel or anti-parallel beta sheets or hairpin loop. And one can simply define motif as a simple folding pattern. So there are two types of motifs, one is the sequence motif, another is the structural motif. Let's discuss sequence motif. A sequence or pattern of nucleotides in a DNA sequence or amino acids in a protein. So here is the specific sequence is given from G, G, T. Okay, so as we have discussed earlier, so there is a specific sequence which is found in this nucleotide. There is at first G, then again T, again T A or T, likewise A C C A, then G T, here is a G T. So such kind of sequence motif, if sequence is conserved in two sequences or multiple sequences, that motif is called as sequence motif. And if the specific structures are conserved in all sequences, that is called as a pattern or protein structure formed by spatial arrangement of amino acids, which is called as beta alpha beta motif or beta mandatory. So beta mandatory is repeat is here, then here is a Greek key where again beta 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 sheets are there, and here is also only repeated beta barrel structures. So significance, so motif mediate protein-protein interactions. Motif is important for the inhibition of potential cancer treatment in numerous oncogenic viruses. So motif also hijacks in some viruses, motif hijacks the cell machinery using modified domains, and there are several disease syndromes related to the disruption of specific drug-mediated interactions which are related with the motifs. Let's discuss domain. Domain can be made from one or more well characterized motifs. So suppose this is a 3D structure, this is a 3D structure of protein, okay, and in this 3D structure, suppose this is a specific motif, this is another motif, okay. So Domain can be made up of one or more motif. If you take this entire protein sequence, so this is the first motif, this is the second motif, and they are independently and they can form a specific domain. So domain can be made up of one or more well characterized motifs which usually occur together and have independent functions. So it is a stable unit of protein, it is a conserved part of a given protein sequence. It decides structure and function of protein, the folded 3D compact structure with stability, and the length of domain varies between 25 amino acids to 500 amino acids. So domain exists in variety of biological context. Similar domains can be found in proteins with different functions. Let's see, this is the SRC homology 3 or SH3 domain, which is the small domain of around 50 amino acid residues that are involved in protein-protein interactions. The SH3 domains have characteristic 3D structure. Okay, so there are types of domain. Domain are alpha category domain, they consist almost alpha helices. Beta category domain, they consist beta sheets and loops. Alpha or beta category domains, they have super secondary structures, that is beta alpha beta motif. Alpha plus beta alpha, that is alpha and beta, they consist of alpha helix beta sheets. So these all classes already we have studied, that is protein folding classes. So what is fold? It is a combination of secondary structure that form the core of domain. Okay, so I'm not going to discuss in detail about this because already we have learned this. So domains occur in diverse range of proteins with different functions. Domains helps for adapter protein, they may act as adapter proteins. Domains may act as phosphatidylcholine. Domain may helpful for phospholipase or for myosins. Okay, so some example we are going to study. So Nck is a cytoplasmic protein which contains multiple SH3 domains. Nck belongs to the adapter family of protein. So we are discussing about adapter proteins. This is the example for this. It involves in transducing signal from growth factor receptor tyrosine kinase to the downstream signal. So if you study the receptor RCK, so RCK domain, so such kind of RCK domain is there. If it is a tyrosine kinase, it is a receptor which is present on the cell membrane. When a specific growth factor is present on this surface, downstream signaling of the receptor tyrosine kinase takes place. So signal is transferred and RASMAP pathway or other pathways are being activated with the help of this domain. So domains function, they have role in binding a particular molecule, they play a role in catalyzing a given reaction. They also together all domains contribute the overall role of protein. So in a single protein there are multiple domains are present which contribute to the overall role of protein. In the next example we will see the domain composition of the enzyme phospholipase D1 and its role. So domain composition of phospholipase D1, it contains three domains, px domain, pH domain and PLD, that is phospholipase D domain. So px domain helps for pi binding, that is phosphatidylinositol binding. pH domain, that is plextrin homology domain, is helpful in the subcellular targeting or targeting enzyme to the particular location. And last, that is catalytic activity is carried out by 2 PLD. So 2 phospholipase D domains are there that have the catalytic activity in the protein. So many domains, these are the many domains, they contribute, as I say there, together all domains contribute to the overall role of the protein. Let's see the significance. Domains are independently stable. Domains can be swapped by genetic engineering between one protein and another to make chimeric proteins. So domains are taken from the single protein and they are used for the making chimeric proteins or hybrid proteins. So also for metabolic engineering one can use the domains by altering protein domains. So for better analysis of protein, which will help in better understanding the different domains causing different functions in a protein. So in short, we will revise domain versus motif. Motif is a super secondary structure, whereas domain is a tertiary structure. Motifs are formed by connected alpha helices and beta sheets through loops, and domains formed by the formation of disulfide bridges, ionic bonds, hydrogen bonds between amino acid side chains. Motifs are mainly have structural function in the protein structure, and domains have many functional importance. Motifs perform similar biological functions through particular protein family, and domain have unique functions. They are not stable independently. Motifs are not stable independently, whereas domains are stable independently. Both have some similarities. They are present in protein molecule. They useful for when classifying protein families. So based on domain or protein, one can classify protein. They both have the structural and functional importance. Moreover, they are made through the interactions of alpha helix and beta sheets. Besides, both are 3D structures. So this is all about domains and motifs. Let's discuss about profiles. Profiles are quantitative discriminators of motif. Quantitative discriminators of motif. How many motifs are there? If single motif is taken, that is studied or stored in the profile database. So profiles are used to model protein families and domains. They provide numerical weights for each position through match or mismatch between a sequence residue and a profile position. So profiles are built by converting multiple sequence alignment position into position specific scoring system. So in the next slide you will study what is profile. If multiple sequences are taken at a single position, what are the matches or what are the mismatches that are stored in the profiles database. So amino acid at each position in the alignment are scored, and according to the frequency with which they occur as represented in the figure. So block substitution matrix, that is BLOSSUM, BLO blocks SU substitution, and matrices can be used to add evolutionary distance weighing these scores. So this is the sequence. So this is the simple sequence. The scoring matrices is used at each position. Okay, so sequence 1, 2, 3, 4, 5 is there. So at this position which amino acids are there, at this position which amino acids are there. Okay, so likewise they are studied and they are stored in the profiles databases. So profile analysis is carried out. So next is the pattern. So pattern is defined as regular expression that describes a short sequence motif with a specific biological function. So it is a regular expression describes motif. Let's study. Okay, so what are patterns? Patterns are many important sequence features such as binding sites or the active sites of enzyme. So if you know binding site or active site, they are found by specific amino acids. In the binding site pockets, there are specific either acidic or basic residues are there, or charged residues are there. Only that residues are present in many amino acids. Okay, either single residue or many residue, and this pattern is conserved with a specific family. So it consists of only a few amino acids that are essential for protein function. Patterns are very good at recognizing such features. They are built by identifying these regions in a multiple sequence alignment. Patterns, the pattern of conservation with the sequence feature is modeled as regular expression as indicated by figure, and ProSite is a database which stores the pattern. So motifs and patterns are stored in ProSite database. You have to remember PPAM is the database which stores the domains, and ProSite is the database which stores motif and the patterns. Now, so this is a motif. We have discussed if entire sequence is considered, it is motif. But in each sequence, what is the regular expression? That is nothing but the pattern. So frequency matrix is used, and the weight matrix. Firstly, the frequency matrix, that is which is repeated continuously, and if the calculation is carried out by using specific matrices, then this data whatever generated is stored in the blocks database. And many motifs, if the repeating many motifs are there, they are stored in the fingerprint database. Okay, so let's talk. This is the motif. So extract the pattern sequence. Here A C is the repeating unit, then again this is the LIH, that is any amino acid. This is the V, which is conserved in every. This is the X4, so 4 amino acids which are repeatedly are present there, and ED. Okay, so ED is the amino acid, either E or D at the end. So this repeated expression then can call it as a pattern. This is a conserved motif used to regular expression. So here any glutamine or aspartate is the ED. So last is the fingerprints. It is a group of ungapped motif, excised from a sequence alignment and used to build a characteristic signature of family membership by means of iterative searching of primary or composite databases. So fingerprints is a ungapped motif which excised from a sequence and used to build characteristic signature of family by searching of primary or composite databases. So single motif methods are good at identifying features in proteins. Most protein families are characterized not by one but by several conserved regions which occur in a certain order. So here we have discussed many motifs are present in these many sequences. So these are nothing but the fingerprints or ungapped motifs. They identifying these regions is the principle behind fingerprints. Fingerprints are composed of multiple short conserved motifs which are drawn from sequence alignment. Each motif is then converted into an individual profile as described in the previous section to create a fingerprint signature. So suppose this is a sequence alignment, you will find there is a one motif, this is another motif, this is another, means these are the conserved regions. Okay, so one can call it as a motif. So there is a specific pattern. That pattern is stored in the profile database. So in profile database, there is a motifs or conserved domains are present, and that all are merged together, and which is called as fingerprint. So fingerprint is nothing but the combination of all the profiles. All the profiles are found by the many motifs. So this means that fingerprints can distinguish individual subfamilies within protein families. This allows functional characterization of sequence at high level of specificity. They identifying individual cellular pathways in which a protein might be involved, the ligand it might bind, the exact reaction may catalyze, and so on. So this is all about the today's lecture. We discussed on today about motif, what are the types of motif, then we have discussed the domain, the types of it, then we have discussed some examples we have taken. We have also studied many motifs play specific role to the protein. Also we have studied the significance of motif. We have studied the difference between motif and domain. We have studied similarities. We have also studied the profiles, which is a database. Which have studied the patterns. Which have studied we have studied the fingerprints. And this is all about today's lecture. So these all are the super secondary structures, or one can also call them as a regular expressions. So based on these regular expressions, the database, or that is secondary protein sequence database, is there, and this is a data. So when two sequences have very less sequence similarity, that is In the next lecture we will study the protein structure prediction based on sequence. There are methods, that is the methods. If more than 50% sequence similarity is there, then pairwise alignment methods are used. But if the sequence similarity is ranging in between 30 to 50%, means many sequences showing 30 to 50% sequence similarity, then the multiple sequence alignment is carried out, and after that one can get these motifs, domains or fingerprints. Okay, so this is all about today's lecture. If you have any doubts or queries, call me back or give me just question, ask me question. Thank you.